Hello everyone, welcome to RKshakti. In today's video, we will be reading chapter 3 from Biology, class 11, Plant Kingdom. In the previous chapter, we looked at the broad classification of living organisms under the system proposed by Whittaker in 1969, wherein he suggested the five kingdom classification viz. Monera, Protista, Fungi, Animalia, Plantae. In this chapter, we will deal in detail with further classification within kingdom Plantae, popularly known as plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and members of the Monera and Protista having cell walls have now been excluded from Plantae, though earlier classifications placed them in the same kingdom. So, the cyanobacteria that are also referred to as blue-green algae are not algae anymore. In this chapter, we will describe algae, bryophytes, pteridophytes, gymnosperms, and angiosperms under Plantae. Let us also look at classification within angiosperms to understand some of the concerns that influence the classification systems. The earliest systems of classification used only gross superficial morphological characters such as habit, color, number, and shapes of leaves, etc. They were based mainly on vegetative characters or on the androecium structure system given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on a few characteristics. Also, the artificial system gave equal weightage to vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by that environment. As against this, natural classification systems developed which were based on natural affinities amongst the organisms and consider not only the external features but also internal features like ultrastructure, anatomy, embryology, and phytochemistry. Such a classification for flowering plants was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification systems based on evolutionary relationships between the various organisms are acceptable. This assumes that organisms belonging to the same taxa have a common ancestor. We now use information from many other sources to help resolve difficulties in classification. This becomes more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Number and codes are assigned to all the characters and the data are then processed. In this way, each character is given equal importance and at the same time, hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information like chromosome number, structure, behavior, and chemotaxonomy, that uses the chemical constituent of the plant to resolve confusions, are also used by taxonomists these days. Algae. Algae are chlorophyll bearing, simple thalloid, autotrophic, and largely aquatic, both freshwater and marine organisms. They occur in a variety of other habitats, moist stones, soils, and wood. Some of them also occur in association with fungi, lichen, and animals, example on sloth bear. The form and size of algae is highly variable, ranging from colonial forms like volvox and the filamentous forms like ulothrix and spirogyra. A few of the marine forms, such as kelps, form massive plant bodies. The algae reproduce by vegetative, asexual, and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by the production of different types of spores, the most common being the zoospores. They are flagellated, motile, and on germination gives rise to new plants. Sexual reproduction takes place through fusion of few gametes. Two gametes. These gametes can be flagellated and similar in size as in Ulothrix, and non-flagellated, non-motile, but similar in size as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size as in species of Eudorina is termed as anisogamous. Fusion between one large non-motile static female gamete and a smaller motile male gamete is termed oogamous. For example, Volvox and Fucus. Algae are useful to man in variety of ways. At least a half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary producers of energy-rich compounds which form the basis of food cycles of all aquatic animals. Many species of Porphyra, Laminaria, and Sargassum are among the 70 species of marine algae used as food. Certain marine brown and red algae produce large amounts of hydrocolloids, that is, water holding substances. For example, algine from brown algae and carrageen from red algae, which are used commercially. Agar, one of the commercial products obtained from Gelidium and Gracilaria, are used to grow microbes and in preparations of ice creams and jellies. Chlorella, a unicellular algae rich in proteins, is used as food supplement even by space travelers. The algae are divided into three main classes: Chlorophyceae, Phaeophyceae, and Rhodophyceae. Chlorophyceae. The members of Chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial, or filamentous. They are usually grass green due to the dominance of pigments chlorophyll A and B. The pigments are localized in definite chloroplasts. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral, or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contain protein besides starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made of an inner layer of cellulose and an outer layer of pectose. Vegetative reproduction usually takes place by fragmentation or by formation of different types of spores. Asexual reproduction is by flagellated zoospores produced in zoosporangia. The sexual reproduction shows considerable variation in the type and formation of sex cells, and it may be isogamous, anisogamous, or oogamous. Some commonly found green algae are Chlamydomonas, Volvox, Ulothrix, Spirogyra, Chara, Phaeophyceae. The members of Phaeophyceae or brown algae are found primarily in marine habitats. They show great variation in size and form. They range from simple branched filamentous forms, ectocarps, to profusely branched forms as represented by kelps, which may reach a height of 100 meters. They possess chlorophyll A, C, carotenoids, and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of xanthophyll pigment. Fucoxanthin present in them. Food is stored as complex carbohydrates, which may be in the form of laminarin or mannitol. The vegetative cells have a cellulosic wall usually covered on the outside by gelatinous coating of algin. The protoplast contains, in addition to plastids, a centrally located vacuole and nucleus. The plant body is usually attached to the substratum by a holdfast and has a stalk. The stipe and leaf-like photosynthetic organ, the frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction in most brown algae is by biflagellated zoospores that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Union of gametes may take place in water or within the ooganium. The gametes are pyriform, that is pear-shaped, and bear two laterally attached flagella. The common forms are ectocarps, dictyota, laminaria, sargassum, and fucus. Rhodophyceae. The members of Rhodophyceae are commonly called red algae because of the predominance of the red pigment or phycoerythrin in their body. Majority of the red algae are marine with greater concentrations found in the warmer areas. They occur in both well-lighted regions close to the surface of water and also at great depths in ocean where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as floridine starch, which is very similar to amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile gametes. Sexual reproduction is oogamous and accompanied by complex post-fertilization developments. The common members are Polysiphonia, Porphyra, Gracilaria, Gelidium. Bryophytes. Bryophytes include the various mosses and liverworts that are found commonly growing in moist shaded areas in the hills. Bryophytes are also called amphibians of the plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid, and shaded localities. They play an important role in plant succession on bare rocks or soil. The plant body of bryophytes is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by unicellular or multicellular rhizoids. They lack true roots, stem, or leaves. They may possess root-like, leaf-like, or stem-like structures. The main plant body of the bryophyte is haploid. It produces gametes, hence is called a gametophyte. The sex organs in bryophytes are multicellular. The male sex organ is called antheridium. They produce biflagellate anthrozoids. The female sex organ is called archegonium, is flask-shaped and produces a single cell. The anthrozoids are released into the water where they come in contact with archegonium. An anthrozoid fuses with the egg to produce the zygote. Zygotes do not undergo reduction division immediately. They produce a multicellular body called sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are of little economic use, but some mosses provide food for herbaceous mammals, birds, and other animals. Species of sphagnum or moss provide peat that have long been used as fuel and as packing material for transshipment of living material because of their capacity to hold water. Mosses, along with lichens, are the first organisms to colonize rocks and hence are of great ecological importance. They decompose rocks, making the substrate suitable for the growth of higher plants. Since mosses form dense mats on the soil, they reduce the impact of falling rain and prevent soil erosion. The bryophytes are divided into liverworts and mosses. Liverworts. The liverworts grow usually in moist, shady habitats such as banks of streams, marshy ground, damp soil, bark of trees, and deep in the woods. The plant body of a liverwort is thalloid. For example, Mercantia. The thallus is dorsiventral and closely oppressed to the substrate. The leafy members have tiny leaf-like appendages in two rows on the stem-like structures. Asexual reproduction in liverworts takes place by fragmentation of thalli or by the formation of specialized structures called gamma. Singular, gamma. Gamma are green multicellular asexual buds which develop in small receptacles called gamma cups located on the thalli. The gamma become detached from the parent body and germinate to form new individuals. During sexual reproduction, male and female sex organs are produced, either on the same or on different thalli. The sporophyte is differentiated into foot, seta, and capsule. After meiosis, spores are produced within the capsule. The spores germinate to form free-living gametophyte. Mosses. The predominant stage of life cycle of moss is a gametophyte which consists of two stages. The first stage is the protonema stage which develops directly from a spore. It is a creeping green branched and frequently filamentous stage. The second stage is leafy stage which develops from the secondary protonema as a lateral bud. They consist of upright, slender axis bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. This stage bears the sex organs. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organs, antheridia and archegonia, are produced at the apex of the leafy shoots. After fertilization, the zygote develops into a sporophyte consisting of a foot, seta, and capsule. The sporophyte in mosses is more elaborate than in liverworts. The capsule contains spores. Spores are formed after meiosis. The meiosis have an elaborate mechanism of spore dispersal. Common examples of mosses are Funaria, Polytrichum, and Sphagnum. Pteridophytes. The pteridophytes include horsetails and ferns. Pteridophytes are used for medicinal purposes and as soil binders. They are also frequently grown as ornamentals. Evolutionary, they are the first terrestrial plants to possess vascular tissues, xylem and phloem. You shall study more about these tissues in chapter 6. The pteridophytes are found in cool, damp, shady places, though some may flourish well in sandy soil conditions. You may recall that in bryophytes, the dominant phases in life cycle is the gametophytic plant body. However, in pteridophytes, The main plant body is a sporophyte, which is differentiated into true root, stem, and leaves. These organs possess a well-differentiated vascular tissues. The leaves in Pteridophyta are small, microphyls, as in Selaginella, or large, macrophylls, as in ferns. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophylls. In some cases, sporophylls may form distinct compact structure called strobili or cones, for example in Selaginella and Equisetum. The sporangia provide spores by meiosis in spore mother cells. The spores germinate to give rise to inconspicuous small but multicellular free-living, mostly photosynthetic thalloid gametophytes called prothallus. These gametophytes require cool, damp, shady places to grow. Because of this specific restricted requirement and the need for water for fertilization, the spread of living pteridophyte is limited and restricted to narrow geographical regions. The gametophytes bear male and female sex organs called antheridia and archegonia respectively. Water is required for transfer of anthrozoids, the male gametes released from the antheridia, to the mouth of archegonia. Fusion of male gamete with the egg present in the archegonium result in the formation of zygote. Zygote thereafter produces a multicellular well-differentiated sporophyte, which is the dominant phase of the pteridophytes. In majority of the pteridophytes, all the spores are of similar kinds. Such plants are called homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, macro, that is large, and micro, that is small spores, are known as heterosporous. The megaspores and microspores germinate and give rise to female and male gametophytes respectively. The female gametophyte in these plants are retained on the parent sporophytes for variable periods. The development of the zygotes in the young embryos takes place within the female gametophytes. This event is precursor to the seed habit, considered an important step in evolution. The pteridophytes are further classified into four classes: Coelopsida, Lycopsida, Sphenopsida, and Pteropsida. Gymnosperms. The gymnosperms, gymnos meaning naked and sperma meaning seeds, are plants in which the ovules are not enclosed by any ovary wall and remain exposed both after and before fertilization. The seeds that develop post-fertilization are not covered, for instance, are naked. Gymnosperms include medium-sized tree or tall trees and shrubs. One of the gymnosperms, the giant redwood tree, Sequoia, is one of the tallest tree species. The roots are generally taproots. Roots in some genera have fungal association in the form of mycorrhiza, for example, Pinus, while in some others, Cycas, small specialized roots called coralloid roots are associated with N2 fixing cyanobacteria. The stems are unbranched in Cycas or branched, for example, Pinus and Cedrus. The leaves may be simple or compound. In Cycas, the pinnate leaves persist for a few years. The leaves in gymnosperms are well adapted to withstand extremes of temperature, humidity, and wind. In conifers, the needle-like leaves reduce the surface area. That thick cuticle and sunken stomata also help to reduce water loss. The gymnosperms are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores are produced within sporangia that are borne on sporophylls, which are arranged spirally along an axis to form legs or compact strobili or cones. The strobili bearing microsporophylls and microsporangia are called microsporangiate or male strobili. The microspores develop into a male gametophytic generation which is highly reduced is confined to a limited number of cells. This reduced gametophyte is called a pollen grain. The development of pollen grains takes place within the microsporangia. The cones bearing megasporophylls with ovules or megasporangia are called macrosporangiate or female strobili. The male or female cones or strobili may be borne on the same tree, that is pinus. However, in cycas, male cones and megasporophylls are borne on different trees. The megaspore mother cell is differentiated from one of the cells of the nucellus. The nucellus is protected by envelopes and the composite structure is called an ovule. The ovules are borne on megasporophylls which may be clustered to form the female cones. The megaspore mother cells divide meiotically to form 4 megaspores. One of the megaspores enclosed within the megasporangium develops into a multicellular female gametophyte that bears 2 or more archegonia or female sex organs. The multicellular female gametophyte is also retained within megasporangium. Unlike bryophytes and pteridophytes, in gymnosperms, the male and female gametophytes do not have an independent free living existence. They remain within the sporangia retained on the sporophytes. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with opening of the ovules borne on megasporophylls. The pollen tube carrying the male gametes grow towards archegonia in the ovules and discharge their contents near the mouth of archegonia. Following fertilization, zygote develops into an embryo and the ovules into seeds. The seeds are not covered. Angiosperms. Unlike the gymnosperms, where the ovules are naked, in the angiosperms or flowering plants, the pollen grains or ovules are developed in specialized structures called flowers. In angiosperms, the seeds are enclosed in fruits. The angiosperms are an exceptionally large group of plants occurring in a wide range of habitats. They range in size from the smallest wolffia to tall trees of eucalyptus. They provide us with food, fodder, fuel, medicines, and several other commercially important products. They are divided into 2 classes, the dicotyledons and monocotyledons. The dicotyledons are characterized by seeds having 2 cotyledons, reticulate venation in leaves, and tetramerous or pentamerous flowers. For instance, having 4 or 5 members in each floral whorls. The monocotyledons, on the other hand, are characterized by single cotyledonous seeds, parallel venation in leaves, and trimerous flowers having 3 members in each floral whorls. The male sex organ in a flower is stamen. Each stamen consists of a slender filament with an anther at the tip. Within the anthers, the pollen mother cell divides by meiosis to produce microspores which matures into pollen grains. The female sex organs in a flower is pistil. Pistil consists of a swollen ovary at its base, a long slender style and stigma. Inside the ovary, ovules are present. Generally, each ovule has a megaspore mother cell that undergoes meiosis to form 4 haploid megaspores. 3 of them degenerate and 1 divide to form the embryo sac. The embryo sac has 3-celled egg apparatus, 1 cell and 2 synergids, 3 antipodal cells and 2 polar nuclei. The polar nuclei eventually fuse to produce a diploid secondary nucleus. Pollen grain after dispersal from the anthers are carried by the wind or various other agencies to the stigma of a pistil. This is termed as pollination. The pollen grains germinate on the stigma and the resulting pollen tubes grow through the tissues of stigma and style and reach the ovule. The pollen tubes enter the embryo sac where the two male gametes are discharged. One of the male gametes fuses with the egg cell syngamy to form zygote. The other male gamete fuses with diploid secondary nucleus to produce triploid primary endosperm nucleus PEN. Because of the occurrence of two fusions, for instance syngamy and triple fusion, this event is termed as double fertilization, an event unique to angiosperms. The zygote developed into an embryo with one or two cotyledons and the PEN develops into endosperm which provides nourishment to the developing embryo. The synergids and antipodals degenerate after fertilization. During these events, the ovules develop into seed and the ovaries develop into fruit. The life cycle of an angiosperm is shown in the figure. Plant life cycles and alternation of generation. In plants, both haploid and diploid cells can divide by mitosis. This ability leads to the formation of different plant bodies, haploid and diploid. The haploid plant body produces gametes by mitosis. This plant body represents a gametophyte. Following fertilization, the zygote also divides by mitosis to produce a diploid sporophytic plant body. Haploid spores are produced by this plant body by meiosis. This in turn divide by mitosis to form a haploid plant body once again. Thus, during the life cycle of any sexually reproducing plant, there is an alternation of generations between gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant groups as well as individuals representing them differ in the following patterns. 1. Sporophytic generation is represented only by the one-celled zygote. There are no free-living sporophytes. Meiosis in the zygote results in the formation of haploid spores. The haploid spores divide mitotically and form the gametophyte. The dominant photosynthetic phase in such plants is the free-living gametophyte. This kind of life cycle is termed as haplontic. Many algae such as volvox, pyrogyra and some species of chlamydomonas represent this pattern. 2. On the other extreme is the type wherein the diploid sporophyte is the dominant photosynthetic independent phase of the plant. The gametophytic phase is represented by the single to few celled haploid gametophyte. This kind of life cycle is termed as diplontic. An alga fucus species represent this pattern. In addition, all seed-bearing plants, for instance gymnosperm and angiosperms, follow this pattern with some variations, wherein the gametophytic phase is few to multicelled. 3. Bryophytes and pteridophytes interestingly exhibit an intermediate condition, haplodiplontic. Both phases are multicellular, however, they differ in their dominant phases. A dominant independent photosynthetic thalloid or erect phase is represented by a haploid gametophyte and it alternates with the short-lived multicellular sporophyte totally or partially dependent on the gametophyte for its anchorage and nutrition. All bryophytes represent this pattern. The diploid sporophyte is represented by a dominant independent photosynthetic vascular plant body. It alternates with multicellular sporophytic or autotrophic, saprophytic or autotrophic, independent but short-lived haploid gametophyte. Such a pattern is known as haplodiplontic life cycle. All pteridophytes exhibit this pattern. Interestingly, while most alga genera are haplontic, Some of them, such as ectocarps, Polysiphonia, kelps, are haplodiplontic. Fucus, an algae, is diplontic. Summary. Plant kingdom includes algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. Algae are chlorophyll-bearing, simple, thalloid, autotrophic, and largely aquatic organisms. Depending on the type of pigment possessed and the type of stored food, algae are classified into three classes, namely chlorophyceae, phaeophyceae, rhodophyceae. Algae usually reproduce vegetatively by fragmentation, asexually by formation of two different types of spores, and sexually by formation of gametes which may show isogamy, anisogamy, or oogamy. Bryophytes are plants which can live in soil but are dependent on water for sexual reproduction. Their plant is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by rhizoids. They possess root-like, leaf-like, and stem-like structures. The bryophytes are divided into liverworts and mosses. The plant body of liverworts is thalloid and dorsiventral, whereas mosses have upright, slender axes bearing spirally arranged leaves. The main plant body of a bryophyte is gamete-producing and is called gametophyte. It bears the male sex organs called antheridia and a female sex organs called archegonia. The male and female gametes produced fuse to form zygote, which produces a multicellular body called sporophyte. It produces haploid spores. The spores are germinate to form gametophytes. In pteridophytes, the main plant is a sporophyte, which is differentiated into true root, stem, and leaves. These organs possess well-differentiated vascular tissue. The sporophytes bear sporangia, which produce spores. The spores germinate to form gametophytes, which require cool, damp places to grow. The gametophytes bear male and female sex organs called antheridia and archegonia, respectively. Water is required for transfer of male gametes to archegonium, where zygote is formed after fertilization. The zygote produces sporophyte. The gymnosperms are the plants in which ovules are not enclosed by any ovary wall. After fertilization, the seeds remain exposed, and therefore these plants are called naked seeded plants. The gymnosperms are produced microspores and megaspores, which are produced in microsporangia and megasporangia borne on the sporophylls. The sporophylls, microsporophylls, and megasporophylls are arranged spirally on axis to form male and female cones, respectively. The pollen grains germinates and pollen tube releases the male gamete into the ovule, where it fuses with the egg cell in archegonia. Following fertilization, the zygote develops into embryo and the ovules into seeds. In angiosperms, the male sex organs, that is stamen, and female sex organs, that is pistil, are born in a flower. Each stamen consists of filament and an anther. The anther produces pollen grains, male gametophyte, after meiosis. The pistil consists of an ovary enclosing one to many ovules. Within the ovule is the female gametophyte or embryo sac, which contains the egg cell. The pollen tube enters the embryo sac, where two male gametes are discharged. One male gamete fuses with the egg cell, that is syngamy, and other fuses with diploid secondary nucleus, that is triple fusion. This phenomenon of two fusion is called double fertilization and is unique to angiosperms. The angiosperms are divided into two classes, the dicotyledons and the monocotyledons. During the life cycle of any sexually reproducing plant, there is alternation of generations between gamete-producing haploid gametophyte and spore-producing diploid sporophyte. However, different plant groups as well as individuals may show different patterns of life cycle, haplontic, diplontic, or intermediate. Thank you for watching. If you like the video,